Bioinformatics Data Mining, Help Wanted, by Brian Baker of Quinnipiac University. With the recent widespread access to the human genome DNA sequence, the new field of bioinformatics has emerged and rapidly expanded. Bioinformatics enables researchers to address the need to store, extract, organize, and interpret this DNA information for the purpose of accelerating academic research and providing commercial uses, such as personalized medicine. As this field has grown exponentially over recent years, this presentation describes how molecular biologists have quickly recognized that a skill gap exists between practitioners with their domain of traditional biological research and practitioners within the domains of computer science and analytics, against the backdrop and context of the history of the bioinformatics discipline. Biologists and computer scientists from various institutions such as the NIH or the National Institute of Health have begun to work together to better formulate a career path for aspiring bioinformaticians. This new discipline incorporates the application of big data mining techniques, statistical analysis, and sophisticated computational algorithms towards the analysis of gene and protein expression and the modeling of gene and protein structure. Rapid developments in molecular research technology within the last few decades have produced a tremendous amount of biological data. This data can only be analyzed in a practical manner by using computationally intensive techniques against large data storage repositories and well-structured annotated databases. The application of statistical and data mining techniques on these huge data sets continues to advance the understanding of biological processes and has evolved into its own field within the domain of biological analysis called bioinformatics. The term bioinformatics was originally coined by Pauline Hogweg and Ben Hesper in 1970 to refer specifically to information processes in biotic systems. However, the term bioinformatics is now used to describe this entire emerging field that cross-pollinates several overlapping disciplines of study, including biology, computer science, mathematics, and engineering. This rich mixture of disciplines provides the means by which modern biological study is conducted to rapidly sequence, align, and annotate genomes, to observe DNA mutations, and compare the evolutionary history between genomes. Bioinformatics is also used to model and predict DNA and RNA expression, and model protein structure and function for the purposes of identifying and understanding, and eventually possibly preventing, the genetic causes of disease. Recognizing how successful populations adapt to new environmental conditions, and determining which genetic predispositions prove to be the most advantageous within those environments. And finally, to categorize biological pathways, networks, and genetic differences among species over time. By the early 1900s, it was known that DNA was a long molecule that consisted of 4 types of bases: adenine A, thymine T, guanine G, and cytosine C. However, not until 1944 was it proven that genes do reside on DNA by Oswald Avery and his colleagues. In 1941, George Beadle and Edward Tatum were the first to identify the role of the gene during their experiments with the bread mold Neurospora. In 1953, the momentous discovery of the double helical structure of DNA was discovered by James Watson and Francis Crick. The double helix provided the key to DNA replication and heredity. Watson and Crick arrived at this conclusion with the help of aligning the DNA structure according to the Chargaff rule, developed by Erwin Chargaff in 1950. The Chargaff rule stated that there existed a 1-to-1 ratio of adenine to thymine and guanine to cytosine content in DNA. In addition, Watson and Crick were aided by the X-ray image observations by Maurice Wilkins and Rosalind Franklin that suggested that DNA is a helical molecule. After manipulating a 3D model using Legos, Watson and Crick realized that the structure resembled a double helix with a parental and child strand that complemented one another and were held together by hydrogen bonds between specific base pairings, A, T, and C, G. Jared Hurwitz and Samuel Weiss later discovered a process called transcription that explains how DNA is used as a template to create RNA. After RNA has been transcribed from DNA, the RNA is spliced into mtRNA, the messenger RNA. This activity occurs within a ribosome outside of the nucleus and within the cell's cytoplasm that synthesizes the protein by reading the DNA code in sequence to form the polypeptide chain of amino acids. Although biologists had known that proteins were composed of amino acids since the early 1900s, it was not until the late 1960s that their conjecture of how DNA was transformed into proteins was confirmed. The transformation of DNA into protein required the DNA code written in a 4-letter alphabet to be grouped into consecutive triplets called codons to satisfy the observed sequence of amino acids and proteins that are written in a 20-letter alphabet. This holistic process, as previously described, is referred to as the central dogma of molecular biology. One of the most pivotal moments in the history of bioinformatics was when Margaret Oakley Dayhoff, professor of Georgetown University Medical Center, compiled one of the first protein sequence databases known as PIR, or Protein Identification Resource, in 1966. Dayhoff pioneered methods of protein sequence alignment and reconstructing their evolutionary histories. Soon thereafter, Needleman and Wunsch developed a program for global sequence alignment. This technique is based on dynamic programming, which attempts to align every residue in every sequence. In 1982, the first DNA database was designed called GenBank from the National Center for Biotechnology Information. Many types of nucleotide databases followed GenBank, including EMBL, European Bioinformatics Institute, DDBJ DNA Databank of Japan, or the National Institute of Genetics, among hundreds and eventually thousands of other DNA databases. The next major development was the development of BLAST, or Basic Local Alignment Search Tool, that greatly aided researchers in finding local similarities between sequences. The Human Genome Project shortly followed and began in 1990 with the collaboration of the U.S. Department of Energy and the National Institutes of Health as a 15-year program to determine the sequence of the complete DNA sequence of the human genome. The first human chromosome was sequenced in 1999. In 2003, the Human Genome Project was completed that sequenced the entire human genome. 3 major databases were utilized for the HGP in different parts of the world, but were interconnected and exchanged data, including GenBank, EMBL, and DDBJ. After biologists had discovered the essential underpinnings of molecular biology in terms of how to sequence DNA and generate predictions for protein expression and function, bioinformaticians developed increasingly more sophisticated alignment algorithms and data mining techniques to interrogate the enormous amount of available genomic data. For example, data mining techniques such as cluster analysis began to be used to compare groups of similar genomic sequences to infer common ancestries. This subdomain of study is called comparative genomics. The complexity of genome evolution has inspired many types of data mining techniques, ranging from heuristics to approximation algorithms to Markov chain to Monte Carlo to Bayesian analysis. Other examples of clustering algorithms applied in gene clustering are k-means clustering, self-organizing maps or SOMs, hierarchical clustering, clustering, or decision trees, and consensus clustering. Many of the algorithms and data mining techniques are focused on homology detection. Homology predicts the function of a given gene sequence by comparing it to a known function of a similar gene sequence. String matching techniques have been widely used for DNA sequencing and homology search to compare similar gene sequences. Perhaps the most efficient and powerful way to detect alignments between two DNA strings is by a technique called dynamic programming, which uses scores based on matching character locations. Two of the most popular and efficient dynamic programming algorithms is fast alignment, or FASTA, and basic local alignment search tool, or BLAST. Protein model homology, in a similar manner, compares protein structure and predicts the structure of other proteins. However, predicting protein structure can be quite difficult. The number of ways a protein structure can fold is astronomical due to the amount of degrees of freedom available, and is considered one of the hardest problems in biology. Several data mining applications for protein pattern detection exist to classify three-dimensional protein structures. In addition to these unsupervised data mining techniques used within high-performance computers, protein folding has been popularized online with protein folding games that encourage players to design new protein structures that may serve to combat or prevent diseases such as HIV/AIDS, cancer, and Alzheimer's. Unsupervised data mining techniques can establish association rules among genetic data, cluster similar gene and protein sequences, and utilize decision trees to classify the genes. Cluster analysis, for example, can be used to analyze a large number of genes simultaneously, called microarrays, that enable molecular biologists to study and group the genes into disease-causing genes from normal genes, as well as study various characteristics of genes under different conditions. Predictive algorithms such as neural networks can be used to predict gene expressions within an organism before they are isolated experimentally. And other types of algorithms, such as genetic algorithms, serve to increase the efficiency of these data mining techniques and minimize the execution time. But regardless of the technique used, these algorithms can only be performed effectively against highly available and scalable databases managed by worldwide institutions. The commercial uses of bioinformatics primarily center on personalized medicine and direct-to-consumer genetic testing. Both have had significant advances in recent years and have had very promising futures from the perspective of financial gain and, more importantly, medical treatment. Until recently, doctors and scientists didn't have the data available to them to determine how different people develop disease and respond to treatments and medications. There was always a uniform approach to medicine, based on broad population averages instead of an individual genetic makeup. The practice of medicine has routinely been reactive in that it's not until the onset of disease that treatments and cures are sought, and unfortunately, in major diseases, the treatments have been imprecise and ineffective. On average, any given prescription drug only works for half of those it is prescribed to. Precision medicine allows for health care that is customized for an individual patient. Doctors are now able to provide better disease prevention, more accurate diagnosis, safer drug prescriptions, and more effective treatments for many of the conditions that are prevalent in our society. Personalized medicine is showing major advances in the areas of addiction, aging, Alzheimer's, cancer, diabetes, microbiome, and rare diseases, according to the Jackson Laboratory, a premier biomedical research institution headquartered in Bar Harbor, Maine. For example, addiction. Researchers have established a strong genetic component for addictive behaviors and are unveiling the genes at work within the brain that control complex behaviors, such as anxiety and addiction. Identifying the biological reasons particular people are at a greater risk for addiction will allow for new prevention techniques as well as treatments. Aging. Increased risk for deadly diseases and difficult-to-manage conditions come with age. Genomic technologies can decipher the changes that occur due to aging to extend health and improve quality of life. Alzheimer's disease. Alzheimer's has two hallmark traits of misfolded proteins that clump together to form plaques and tangles in the brain, disrupting nerve cell function. Jackson Laboratory is attempting to identify the complex genetic deviations and different genes all interacting and conspiring to lead to the disease. Cancer. Computational biology approaches are being used to analyze genomic data sets from patients with brain tumors to understand therapy resistance. Profiling individual cancers will lead to tailored treatments for each patient so that the cancer can be targeted without harming the patient and even prevent cancer from developing altogether. Diabetes. At the Jackson Laboratory, the development of pancreatic islets, which produce insulin and other metabolic hormones, are being explored, as are conditions that alter the genetic programming of islets and lead to abnormal functions, such as insulin resistance. They believe that there is the possibility of producing healthy pancreatic islet cells from pluripotent stem cells or other kinds of fully developed cells in the future. The forefront companies in direct-to-consumer genetic testing are Ancestry.com and 23andMe. Ancestry.com. To enhance its ability to provide accurate genealogy information, Ancestry.com will also sequence an individual's DNA, focusing on 700,000 markers important to determining one's race, lineage, and other factors. The DNA service has over 7 million customers and can provide users their race and what country they're from, and also connect them with other relatives who share a DNA profile. Ancestry.com is investing between $10 and $15 million into their DNA service and have bioinformatics scientists on staff trying to scale algorithms designed to handle hundreds of samples to work with hundreds of thousands of samples as the service increases in popularity. 23andMe. Named for the 23 pairs of chromosomes in a normal human cell, 23andMe is the only genetic service that provides results directly to the consumer that are FDA approved. A customer is able to download the raw genomic data and use information from other sites to interpret the results. 23andMe have researchers who study their aggregated customer data for inherited disorders. They also, however, partner with academics and pharmaceutical and biotechnology companies and have acquired a startup called Cure Together, a crowdsourced treatment ratings website with data on over 600 medical conditions. As the bioinformatics discipline has grown exponentially over recent years, molecular biologists and educators have quickly recognized that a skills gap exists between practitioners within their domain of traditional biological research and practitioners within the domains of computer science and analytics. Within the last decade, according to Kristen Sanani, PhD, Associate Professor with Health Research and Policy at Stanford University, undergraduate and PhD programs have increased to about 80% and MS programs to 60%, which impresses upon educators the necessity to keep the curriculum relevant. Biomedical Computation Review in 2015 has assessed the current educational landscape and have related how biologists, computer scientists, and educators from various institutions and universities have begun to work together to better formulate a career path for aspiring bioinformaticians. Universities working interdepartmentally and with bioinformatics departments are developing updated and relevant curricula focused on defining core competencies such as biology, statistics, and computer science, while remaining flexible enough to incorporate the ever-evolving technologies of big data platforms and analytics. Bioinformatics educators are confronting this challenge of binding together a coherent and interdisciplinary curriculum through several fronts. Educators are experimenting with ways to bridge the gap between students and researchers with disparate training backgrounds by developing new courses that supplement traditional undergraduate coursework, merging divisions between departments into new ones for bioinformatics, encouraging biology students to cross-train with computer science students, and sponsoring and fostering new bioinformatics organizations that facilitate the training of students and researchers. To help reduce the heterogeneity of scholastic backgrounds, educators have launched several initiatives and consortiums. In 2012, educators banded together and founded the Global Organization for Bioinformatics, Learning, Education, and Training, or GOBL, to address the need to establish global curriculum standards and accreditations, and to share high-quality training materials to prepare students for the job market. The International Society for Computational Biology, or ISCB, surveyed both commercial enterprises, universities, and job listings to discover that there existed a large discrepancy between what skill sets possessed and what skill sets employers expected from new hires. The ISCB published their curriculum recommendations in PLOS, Computational Biology, in 2014. As a result of the ISCB report and responses from interviewees from the Biomedical Computation Review in 2015, several areas in which training programs are failing to prepare future bioinformaticians were highlighted. Some of these areas include the lack of bioinformatics training in big data statistics, which can be quite different than what traditional statistics courses offer. Engineering skills that are required to build robust, reproducible tools, and the lack of exposure for computer science students to wet lab experiences. It was also mentioned the need for capstone courses that combine biology and computer science together, which requires institutions to hire bioinformatics teachers. Despite the call for homogenizing the curriculum to a large degree, the ISCB also recognized the need to tailor the curricula to the end goals of the student. The task force outlined three distinct groups of bioinformatics practitioners that must be considered separately to address their unique perspectives and training needs. The three groups consist of bioinformatics users who are biologists or physicians who need bioinformatic tools to conduct research or patient care, bioinformatics scientists who develop algorithms and tools intended to answer biomedical questions, and bioinformatics engineers who build software and computational infrastructure for the purposes of enabling bioinformaticians to work more productively. Each of these groups will vary in their depth and breadth of knowledge in different aspects of bioinformatics, such as life sciences, software engineering, and big data platforms. Perhaps the most prevalent response from educators across the board, according to the survey from the Biomedical Computation Review, was the importance of emphasizing the need to adapt to big data. With the phenomenal growth in databases that contain terabytes of genomic data, it is imperative that biology students are well equipped to manage this data by becoming well acquainted with machine learning, multiple hypothesis testing, detecting bias and confounding data, cleansing and transforming data, and performing the correct inferences and predictions by accounting for errors within the data. The National Institute of Health, or the NIH, one of the premier health institutions within the United States, is actively helping to prepare the national workforce in biomedical science and big data through their Big Data to Knowledge, or BD2K initiative. The major goal of this initiative is to narrow the gap between needed and existing biomedical data science skills. The BD2K has and continues to support biomedical students and researchers by creatively utilizing existing mechanisms to provide outreach to thousands of biomedical scientists. Since 2017, the portfolio has grown close to $15 million, consisting of 71 grants and awards. Also, 26 administrative supplements were issued in fiscal year 2016 to supplement existing activities and training opportunities for all skill and career levels, totaling $2.7 million. Beyond the traditional classroom and curriculum recommendations, bioinformatics practitioners and students can take advantage of public online courses and repositories of molecular data. The EMBL-EMBI maintains a freely available molecular database, for example, that can be queried and analyzed using their comprehensive set of tools. Online courses are also freely offered through venues such as Roslin, the Swiss Institute of Bioinformatics, and bioinformatics.org. These online courses span the gamut, including courses specifically for biologists, including Perl, R, MySQL, MATLAB, Python, image processing, gene expression analysis, SNMP prototyping, and microarray data analysis, among others. Besides the individual courses, these institutions provide boot camps that generally last a couple of months dedicated to bridging the gap between biologists and computer scientists. MOOCs, or massive open online courses, such as Coursera, edX, Udacity, and others, provide top-notch introductions to bioinformatics. These online courses can serve as a complement to formal study at a university, or provide existing bioinformatic practitioners a means by which they can keep their skills current and relevant. A career in bioinformatics has a promising future and can be attributed to the tremendous growth in available biological data, the steady advancement in big data technology and methods, and the level of commitment shown across a wide spectrum of institutions, including universities, organizations, and online resource providers. Academic frameworks have been developed to strengthen core competencies and remove barriers to entry for audiences with different academic and professional backgrounds. With this increased support, the recognized skills gap between research biologists, computer scientists, and engineers, and data mining specialists have begun to narrow. Educators have benefited greatly from these task force initiatives and public engagements between practitioners who have offered their unique perspectives and experiences. These interactions between subject matter experts have ultimately produced a well-rounded foundational curriculum to prepare prospective bioinformaticians for a successful career. However, as the history of biological research attests, those who are the most eager to expand their education and remain astute with the latest technologies and data mining techniques will be the most capable to capitalize on this dynamic and growing field. The opportunities to better understand biological processes and advance personalized medicine has never been greater than today. The fulfillment of these hopes to prevent and eradicate disease and improve the human condition rests largely on encouraging and properly educating future bioinformaticians.